striving to be a voice for waterbirds in crisis. The goal tonight is to actually make something that we think is not being talked about enough, avian influenza, also known as bird flu, high path avian influenza, kind of give you some of the inside details and ways that you can interact and things you should be aware of, and some of the dynamics along the way. The main speaker tonight will be our veterinarian and director of research, Rebecca Duerr. I am here almost as the Becky Whisperer, Dr. Becky Whisperer. She is going to handle the very scientific side, and when things are a little complex, I might jump in and make sure that the key things land. Feel free to send your Q&A questions as the thing goes. You're welcome to use the chat window as well. We'll try to answer things as we go. As I said, I'm trying to be the voice that keeps it simple. One detail I wanted to point out is that if you've heard anything about this in the news, it is likely in connection to the chickens and eggs that we consume as people. In a lot of ways, that part of the equation of bird flu has gotten more coverage and actually dominates in the government agencies. So the USDA has more say than perhaps how the U.S. Fish and Wildlife Service. There's a lot of interest in and concern over the impact on wildlife, but the discourse really is very much about chickens and eggs that we eat. So here's our hope to help out. Many thanks to colleagues who have helped share their best knowledge with us, and we are doing what we can to share it all with you. Thanks, and Dr. Becky, please go when you're ready.
Thanks, JD. Can you hear me okay?
You sound great.
Okay, great. Okay, yeah, welcome. Thanks everybody for coming. Appreciate your interest in this topic. It's been on the top of my mind for the last year or so. So barring any technical glitches, let's go. Here we go. Mostly I thought with this talk tonight, we thought we'd do a kind of a state of the outbreak talk. We have done town hall type meetings with our own volunteers, many of whom are attending tonight prior to this, but haven't really done anything intended for the general public. We do have some general public folks listening tonight, in addition to some veterinarians from far, far away, including, I think David Roberts has signed up to be here. He's the vet down at Sandcobb in South Africa, and some folks from Australia. So we'll see who actually makes it tonight. All right, here we go. All right, first a little background, and tolerate my Halloween slide. Okay, so for those of you who haven't taken a biology class in a very long time, which, you know, is a whole lot of us, I thought I'd just throw at least some basic information here about the nature of viruses, since they've certainly been in the news a lot since the nightmare of COVID and all of that. So we typically deal with 4 different kinds of pathogens. There are a few others I'm not going to bother mentioning, but you know, the largest of those are the parasitic worms. And some of those we could see with the naked eye. And then there are also some single-celled organisms that are parasites, like protozoal organisms that are also can be a big deal in some of the animals that we deal with, like your dogs and your cats may have some big problems with those, or you could yourself, like if you had a case of Giardia, that would count. There are also a lot of pathogenic bacteria. These are all getting smaller and smaller as we go down. So protozoa and bacteria you can see with a regular old microscope, and we can study those that way. But viruses are weirdos in that they're extremely tiny organisms, and they're not even really organisms because they're not technically alive. So they strictly harness the molecular apparatuses of the organisms that they infect in order to go about their own business. So they're nasty little things. All right, so here's more, but here is a whole bunch of different viruses that are of importance to humans, kind of classified by whether they have DNA as a genetic material or RNA. And here's a selection of a whole bunch of viruses of concern to humans, from herpes. Smallpox is a giant virus that's almost visible with a regular old microscope. But there's also Ebola. Those are weird, really weird shaped viruses. Rabies is another weird one that's shaped kind of like a bullet. Coronaviruses, of course, we're all a little more familiar with, but West Nile virus, Zika virus, those guys are down in this group called flaviviruses. Those are often injected into people by mosquitoes. And then there's also some other viruses like polio on here. So it's just kind of a selection of viruses. So here are the influenza viruses in family Orthomyxoviridae. So these viruses have 6 to 8 strands of genetic material in them, and they have this protein coat called an envelope on them. And one of the important things about having an envelope when you're a virus from the point of view of humans is it makes you a little more susceptible to disinfectants. So that's actually really good news for that. Viruses that do not have an envelope can be extremely hard to kill and stop from infecting people. Like foot and mouth disease is down there with polio in the coronaviruses, and those guys don't have an envelope on them, and consequently, that can cause massive outbreaks in cows and cause us to, the only thing that will kill them is pretty much fire. So anyway, so having an envelope is a good thing when we're dealing with some of these viruses. There are at least 4 types of influenza viruses. They are parsed out into A, B, C, and D types. A, which may or may not be related to the fact that they're avian, tend to cause seasonal flus in humans. There are also some other ones like swine flu. These viruses are really, really commonly developing new strains, which is one of the reasons why our flu shots are always different every year. And I'm going to talk about those rather a lot more. So influenza A viruses have a really complicated classification. And you've probably heard of the H's and the N's. Those are 2 different kinds of surface proteins that they have. So they're hemagglutinins, which is a protein, and neuraminidase, which is another surface protein. So there are 18 types of H's and 11 types of N's so far. And they mix and match and make all sorts of different combinations. And they are constantly undergoing differentiation into new strains, which is really difficult and why we often are frustrated with our flu vaccines, because like, I didn't get the strain this year. That's because these things change constantly. And they can even accumulate differences that are important just moving between animals. But primarily, a lot of these changes that they undergo are usually related mostly to the H hemagglutinin portion of their protein coat. These viruses are also characterized by how pathogenic they are to poultry, and that's where the poultry angle comes in. So the viruses that have an H5 or an H7 are the ones that sometimes become highly pathogenic to poultry and are very lethal to chickens and can roll right through a chicken house and kill hundreds of thousands of chickens really fast. So these are, of course, extremely important to human food chain, which is probably ultimately why our governments pay more attention to them than to wildlife. So these influenza A viruses have been responsible for a few human influenza pandemics in history. So including the really big one in 1918 was an H1N1. There have been a couple of others over time. And those influenza A viruses not only are characterized by that A, but also they have subtypes. And then below that, there's clades and there's subclades, and there's even more distinctions in regard to the exact genetic codes that they have in those protein coats. So the naming of these things can get pretty complicated. If you have a dog, you may have had their dog vaccinated for canine influenza. That's an H3N8 or H3N2. There's also a cat influenza. That's an H7N2. And then there are a couple of these that only really affect bats. The higher numbers of both the H's and N's are strictly bats, and hopefully they'll stay just bats. And so I thought I also might throw on here, this is generally how these things are named. So they're named by the type of virus and where they were isolated, strain number, virus type, et cetera. So the naming can get kind of complicated. All right, so for avian influenza viruses, and remember, it's a whole class of viruses. It's not one particular thing. The low pathogenic strains So your average generic rando virus out there in the wild, circulating among wild animals, is likely a low pathogenic strain. The problem comes when those strains get transferred into human agriculture. So when wild animals have contact with our domestic animals, which are often genetically similar, held in overcrowded circumstances, et cetera, these viruses can morph into something that's hazardous. And then humans can potentially get exposed by our agricultural practices or at wet markets. Certainly in a lot of parts of the world, refrigeration is not that common. So a lot of times animals are kept live and sold to their end consumer as a live animal, and then they're slaughtered right before they're eaten. And that negates the need for refrigeration. So it's totally understandable. But when these viruses have mutated into something hazardous, then they can go back into wildlife, or they could potentially affect people. So that's kind of where we are today. So these viruses also, influenza A viruses, survive better in the environment away from animals. So the animals can poop them out or have them contaminate water with them just by being in the water. And the virus will survive better and remain more infectious at lower temperatures versus high temperatures, fresher versus salty water, wet versus dry environments. And then some other things can also affect them, like the pH or how acidic the water is. So these viruses tend to be cool, freshwater, wet environments are where the viruses last longer. If you're in a hot desert where it's dry and salty, they don't survive outside the bird much longer or very long at all. So they can be inactivated by high temperatures or low pHs. And one of the problems with them in the environment is that they can persist in the environment, in the water, for quite a while if it's cold and fresh water, like in a freshwater marsh, say, where the water's hovering around freezing. They can last quite a while out there. Whereas in the hot desert, they don't last very long at all. They can also continue to be viable infectious organisms on non-porous surfaces or fabrics, that sort of thing. And it's a little bit different on each type of material. So historically, responses to highly pathogenic avian influenza outbreaks in the United States have been largely a poultry problem. So what would happen for the last many, many decades, 100 years, is that a particular farm would be found to have a problem, like maybe a bunch of their chickens are dying in a big mess. So the response of the governmental agencies would be to come in there and cull all those animals and quarantine the premises. So those animals would all be killed. The farm would be cleaned and disinfected. There would be a buffer zone set up where no animals are allowed to move in and around the farm. So there'd be a buffer zone around, and then a surveillance zone where the USDA, so the United States Department of Agriculture, would come in and they would be diagnosing what the problem is. And if it's one of these H5 or H7 subtype viruses, then they'd be establishing all this regional control to prevent that virus from spreading. And then once the property is free of disease, everything gets clean and disinfected and negatives, then that farm could get restocked with another round of animals and kind of start over with undiseased animals. So that's the kind of the way it went for a very, very long time. And typically these viruses, the low pathogenic versions that were out in the wild, didn't really affect wild animals too terribly much. But why is this virus such a big deal? And why is it making our eggs so expensive? So I'm not going to talk about poultry a whole heck of a lot, other than to say that these current outbreaks of the various strains of this virus that are going around have caused massive problems in the poultry industry, almost worldwide. So far, Australia and New Zealand, Antarctica are about the only places that are not being affected by our current outbreaks of this virus. But hopefully they won't join us soon, but hold my breath. So why is this virus such a big deal? Here we go. Here is a timeline of these highly pathogenic avian influenza virus H5 type in non-poultry birds. So there was a little outbreak in 1961 in South Africa that was thought to have been a virus that came out of poultry and went in and killed a bunch of terns. But then nothing really more happened for a long time. So then in 1996, a particular strain that's now called the goose guangdong strain of H5 came into existence in Hong Kong around 96. And there were some little outbreaks over the next decade or so in China. But then things started to heat up a bit. And by 2010, a new clade of this goose guangdong virus started to emerge called the 2344 clade. And then that really started to take off. There was a little incursion into the Western United States from Asia in 2014, which thankfully didn't really do a whole heck of a lot outside the Pacific Northwest. But then it really took off in Africa and Europe and Asia over the course of the next few years, 2015 to 2018 or so. And then in 2020, 2344 became goose guangdong 2344B. And that one came into existence and started spreading around. So here's what's happened since then. This is all extremely unpleasant. Sorry. It's unfortunately the reality of the world. Okay, so 2020, 2344B emerged. And then we started seeing problems for serious and real in a lot of the wild animals. Great skuas were one of the first species that started having huge problems with birds dying at breeding colonies in Europe. And then it entered into North America in December of 2021. I'll show you a map in a minute. And then we started seeing problems here, but it was also still causing problems everywhere else. So there are eider chicks in Canada, all sorts of gannets and murres were affected in Eastern Canada on the Atlantic that following summer. Meanwhile, there's cranes and geese, and South Africa is dealing with Cape cormorants and a whole lot of other endangered seabirds like African penguins dying when they were already having population problems prior to this disease hitting. And it finally hit California about the same time. It was causing massive mortalities in one of the Caspian tern colonies on Lake Michigan, killing pelicans and geese. And then it hit South America shortly after it hit California. Although oddly, it seems to have entered South America primarily on the flyway from the Midwestern North America. It didn't come down the Pacific coast and then travel south. It went south first. So once it hit South America in around October 2022, there were massive reports of Peruvian pelicans dying, blue-footed boobies, all sorts of other endangered birds like the Humboldt penguin. And it's been kind of wreaking havoc ever since. So there are all these different populations of birds all over the world, different species, and different amounts of dead animals. And unfortunately, a lot of illnesses, when they roll through a breeding colony of animals, they often will kill the young of the year. And, chicks and baby animals are very sensitive to diseases. The big deal with this disease is that it's killing breeding adults and their chicks. So that is one of the biggest differences of why this disease is a particularly huge deal worldwide for bird conservation. So you probably heard that there were a whole bunch of California condors that were affected. In Arizona, we haven't had that problem among the California condors in California yet, but it certainly is being planned for. But meanwhile, there aren't very many. There's only 500 some California condors in existence, but 21 of them died pretty quickly. So there were some big concerns there. Whereas there were 30,000 black-headed gulls and Arctic terns and breeding colonies in the United Kingdom. And more than 500,000 seabirds are thought to have died in Peru and Chile, according to many of the papers that are now starting to come out about this. More locally, we have had a big die-off in Puget Sound in the Columbia River this past summer, like only 2 months ago. Caspian terns, Puget Sound over on the Olympic Peninsula. And then very recently, also there was a small die-off of common murres up in really remote Alaska on the Bering Sea. And of course, if humans don't notice that there's a bunch of dead animals, then they don't get counted, that's for sure. So here's kind of the way it came into the United States and North America in general. So it came in from Europe and then promptly started spreading. So these maps were being made by the United States Geological Survey early on in the event. And I later found out that there was just one guy making these maps, which is why they've stopped being made, because he was having to hand plot every one of these dots. So the red are commercial poultry, the yellow are non-commercial poultry, backyard chickens, essentially. And then the green were wild birds. So here we are in April 2022. California, we're starting to get nervous. Minnesota was getting it really hard. The wildlife centers there were very heavily impacted. At one point, the Minnesota Raptor Center had 40% of the animals that were coming into their door were positive for this disease. By June, it's in Oregon and Washington, Alaska. We're still getting more nervous in California. There's an odd green dot there in Arizona, which was 4 neotropical cormorants. Oddly, neotropical cormorants apparently breed in Phoenix, middle of the desert, in a gated community with a pond. Who knew? Anyway, so we're definitely getting more and more nervous about this. And then, of course, during this time period, we also started to find this disease happening, being found in mammals. There were some seals up in Maine, a whole lot of foxes all across the United States and Canada, those little blue triangles or diamonds, and also some skunks and raccoons. So it started to hit some mammals, which certainly makes people more nervous. This is what that breeding colony on Lake Michigan looked like. So those are adults, mature breeding adults that are dead and being picked up by a person in PPE and carried away. So one of the ways that management agencies have been dealing with this is actually picking up the carcasses, because it prevents predators from eating them. And it also should cut down on the amount of virus in the environment, like picking up the dead animals. So that is something that happens. So here we are by July. Now, okay, we finally had some in California. July 15th, 2022 was the first detection. It was up at in the Sacramento Valley at Sacramento National Wildlife Refuge in American white pelicans and Canada geese. And then by October, Northern California was all full of it. And this was about the same time it was hitting South America. So here we are. South America, and this video was sent to me by a horrified Peruvian vet that I know from UC Davis. These are dead Peruvian pelicans on the beach. Yeah, sorry, it's horrifying. Okay, so by November, we finally had it in Southern California as well. So those of you who are really familiar with us know that we have a center in the San Francisco Bay Area, which is kind of midway up California. And we have another center down in Los Angeles. So now both of our centers were involved in areas that definitely had the virus present by the end of November. So who's paying attention to this in the United States? There are 2 government agencies. Oddly, neither of them are the U.S. Fish and Wildlife Service. One of them is the United States Geological Survey, who used to be making all those great maps. They have a program called the WHISPERS Event Reporting System. And this is a system where governmental wildlife people and like game and fish managers can report events and have them kept on this one database. The trouble is it's voluntary reporting and it's not used all that much. So there are rather a lot of events that those of us who've been listening to this and talking to other biology folks through social media and such, there's a lot of events that we're aware of, especially in the Western United States, that are not here. But those 2 gold dots up in Alaska there are represent those 400 MERS. And then that red dot north of them is a single red fox. So overall, just for the United States, not looking at Canada or Mexico or any other nearby areas, this program, if you search for it, you can get 32,508 affected animals. And one of the nice things about this program is if everybody uses it, it would be awesome. All those folks who are managing disease outbreaks and dealing with wildlife. You can do a search on here. Anyone can. It's publicly available. You just stick your time range on there, what you're searching for. And if you click report, it'll generate a PDF for you of all the reports of that particular thing that you're interested in. So it's a pretty cool system, but unfortunately, it's not being used all that much. It's not being used enough, in my opinion. There's another one that's put out by USDA. Of course, these are the folks concerned about food supply. So primarily their deal is poultry. But they do have a page on wild bird avian influenza surveillance. The trouble is that it seems to like they're only updating it maybe quarterly or something. It doesn't change very often. And there. This is only their own USDA disease surveillance information. It's not collecting information from any of the other organizations that are looking at this. So it's also extremely incomplete information. But it looks like they were trying to model it kind of on the COVID dashboard that whatever that university was that made that for COVID, where you could look at the whole world and zoom in on areas and see how it is in your area. Anyway, so those are the two main governmental agencies that are tracking any of this stuff where you can pay attention online, at least for the United States. As far as worldwide, there are a few UN-based organizations like the FAO, Food and Agriculture Organization, and a couple of others that are paying attention somewhat, but nobody really has a clear idea on exactly what's going on with which species of wild birds. So the USDA's website is pretty handy. However, it has some serious limitations. So this is their primary area where they're tracking this particular disease. And you can go on there and, again, publicly available. You can download all of their animal cases. So here's the most recent cases that we've had in California. So you can see it's been 2 months since we had any positive cases tested. But of course, all of this is limited by who's doing the testing and who's sending in animals, all that sort of stuff. It doesn't tell you anything about how many tests are run, who's doing any sort of screening, all that sort of stuff. But most recently, it's been mostly raptors and a gull, Monterey, kind of in the middle of the state here. So I downloaded all that information for the whole time period since this virus has been a problem. And I get this. So the USDA site separates things by how the animals were encountered. So were they dead in the wild? Were they hunted? If people are hunting waterfowl, a lot of times governmental agency folks will be out there swabbing birds that are killed by people for food. And those animals, tests will be listed as a hunter harvest and sometimes listed as a live bird. There's a tiny little bit on here that are agency harvest in that kind of pea green. So the teal are live birds, blue's hunter harvest, and then the purple is animals that were sickly or dead in the wild. So as you can see, there's rather a lot of, there's some interesting differences here. And even though this data is limited, it's the best data that we have. So you notice that these birds that are hunter harvested were not sick in the wild. These were animals that were probably shot while they were flying, so they were probably not that sick. And these are, in particular, almost exclusively dabbling ducks. So we got teals and mallards, widgeons, gadwalls, shovelers, pintails, etc. There are a couple of other species that are a little more confusing, like wood ducks. They have a certain amount that appear to become sickly and die in the wild. and also some that are routinely caught by hunters and swab as positive. Snow geese also have some of both worlds there for them, but largely those animals were sick and dead in the wild. So unlike the dabbling ducks, the geese seem to be very susceptible to this disease. This graph only includes the top 20 species with positive tests. Over time, these numbers change, but there are several species on here that we certainly treat at International Bird Rescue, namely, largely most of them, other than the hawks. We don't do vultures either, but we do pretty much most of the rest. So these are our patients, so that's in particular why this disease has been such a big deal to us. We also have animals in our care routinely that are highly susceptible to the disease. They're going to get sick and die, and have birds that might be asymptomatic carriers of the disease. So they could be a risk to the other birds we have in the center. So those are some of our challenges dealing with these animals. So here's another way of looking at those species. And one thing to really remember about the USDA numbers is that the number of confirmed tests do not at all reflect the number of the affected animals. If there are 2,000 dead geese, it's likely only 2 or 3 of them are going to get tested. So these animals are all proxies for an unknown number of other animals, except maybe the California condors, since every single one of them was getting tested. So we have those 2 federal agencies, and then the state and local agency response, depending on what state you're in and what areas you're in, have been extremely varied as a response to this disease. And unfortunately, among the wildlife community, Facebook and email listservs have been extremely important in trying to figure out what the heck is going on where. So as you can see in this graph, the dabbling ducks have been, I parsed them out just because they are so much that they appear to be asymptomatic carriers of the disease, whereas some of those other waterfowl, like the geese and the swans, appear to be pretty susceptible to the disease. And then diving ducks can go either way. And mostly that's represented by the wood ducks. We also have a lot of sea ducks in the world, such as, eiders and scoters and those kind of guys. They appear to be pretty susceptible to the disease. Most of the raptors are quite susceptible. And then historically, shorebirds and gulls are thought to be asymptomatic carriers of the disease, but at least so far in the United States, it appears that they're pretty susceptible to it. Although I actually don't know whether anyone is randomly sampling gulls out there to see if they are spreading it around. But we certainly know some of our shorebirds, including some that are threatened and endangered, are susceptible to disease, such as western snowy plovers are susceptible, and there are some deaths that have happened, even though there's only 2,500 of our western snowy plovers left. A couple of them have died of this disease. And then pelicans, our California brown pelicans were delisted from the endangered species list in 2009, and pelicans all over the world have certainly been quite susceptible to the disease. And who else? I lumped all the long-legged wading birds in here. There's all sorts of birds in that group, quite diverse: herons and egrets and flamingos and cranes and all sorts of long-legged animals. Passerines. So passerines are definitely, so our songbirds are a different issue in many ways. There haven't been very many positive tests, but there has been a fair diversity of animals that have tested positive, including quite a few crows and ravens appear to be susceptible to the disease. One of the issues with passerines, of course, though, is that, if a whole lot of passerines die, if they don't land on the road, a lot of times nobody might notice. And there's probably a substantial under-testing of passerines going on. So don't be surprised if it becomes more of a big deal in passerines. However, many of these other species of birds, especially the seabirds, tend to breed in huge colonies, and that seems to be where they end up with huge mortality events. whereas a lot of passerines don't congregate like that when they're breeding. So that probably is a benefit, although it certainly could be a problem in, say, cliff swallows or some of the species that do nest in large conglomerations of nests. So I guess as things progress, we'll see what happens. I should probably mention mammals. So this is the positive tests regarding mammals in the United States as of yesterday. So as you can see, they're running out of symbols for different types of mammals. So we're naming individual species here. Obviously can't do that with birds since there's so many different species. But there have been a lot of foxes and a lot of skunks that have been positive. Those are the purple circles and the gray diamonds. Of interest on the West Coast here, we recently, just this past month, had positive harbor seals. Those are the first positive marine mammals on the West Coast since the outbreak in Maine happened in similar species almost a year ago now. There's been one dolphin in Florida, and we'll see what happens on the West Coast here. Bears have been an issue. There have been cases in grizzlies and black bears and mountain lions. And some of these animals are only getting found if they have a collar on. Like I think one of the mountain lions in California only really got identified as having this disease and dying of it because it had a collar on. And, you know, not that many animals have collars. Okay, that's all I'm going to say about mammals. Of course, with mammals, being a human, there are concerns that this disease could move to humans. There have been cases in, okay, it's not all I'm going to say about mammals. There have been some cases in cats in Poland, mink farms. Who else? There's concerns about some of the sea lions in South America have had some concerns about animal to animal spread. So at the current time, it's thought to be low risk, but possible that this virus could end up mutating to spread among mammals. And hopefully it's not going to do that. So anyway, so that's the rough worldwide situation that we've been dealing with. And so how have wildlife centers been dealing with this? Since we're, front lines, we get live animals that are sick. We don't know what's wrong with them, with limited ability to diagnose whatever their problems are. How have we all been dealing with this? Okay, well, in the case of our clinics, we've been writing lots of protocols and implementing them. And it's been an evolution. We did finally combine all of our protocols into one great big protocol for different areas. So we essentially had to divide things out into a screening area where we're looking at that animal before anything else happens and trying to figure out whether the animal is an infection risk. And then if the animal makes it through that judgment period, they go into a quarantine area, and that allows us to see how is that animal when it's warm and well hydrated. Most of the animals that we get are cold, they're emaciated, they're generally have lots of problems, they may have injuries, etc. So they're not in really good condition to really judge what their condition is until we get them a little more stable. And then in our regular clinic area, what would happen if we had an animal break with this disease while it was out in an aviary that had 60 other animals in it? So we've had to consider all these different scenarios and try to figure out what we're going to do. So at most wildlife centers, the primary goal is to keep the virus out of the hospital. And unfortunately, that has meant probably euthanizing some animals that did not have the disease. Considering that in the United States, at least, the animals that are positive for this disease have largely had neurologic signs. We'll talk more about their clinical signs in a minute. We have been triaging out neurologic birds before they enter our general care areas. And that has meant euthanizing animals that have symptoms that could be avian influenza before bringing them into the clinic. And then even separating out, do you have to walk through the facility in order to go put that animal in a freezer after it's been euthanized. So at one of our centers, we had to purchase an extra freezer just to keep outdoors. So we did not have to traipse through the clinic in order to get to the freezer. So there are a lot of pathways through the hospital that we really had to consider. We've also had to do personal protective gear for all new bird exams because of the possible human infection risk of these animals. And then we have been instituting quarantine periods as well as we can, given our facility limitations, facility and staff limitations, and having foot traffic control. So most centers have changed the way they move through their center and might have external buildings or little temporary sheds or even tents. Tents haven't held up very well long term. So most wildlife centers have gone to having an outbuilding or something like that where you can do an initial exam on animals. And then having these foot traffic controls of the way people move through your facility to try to reduce the spread of pathogens through the building. And then only birds that have been cleared through screening and quarantine are allowed in those general care areas. We've been trying to keep animal groupings as separate as possible, in particular, keeping those dabbling ducks as far away from other species as possible. Pardon me. Sorry. And having dedicated clothing for our staff volunteers, and especially if they have pets and poultry at home. Okay. All right, so it generally goes this way. You screen new animals. Are they neurologic? If so, they might get euthanized unless we have a reason that they might have neurologic signs like botulism, which we're currently dealing with, or they perhaps a victim of a harmful algal bloom or a head trauma. With those animals, we'll keep them in quarantine and test them. If they're not neurologic, then they get quarantined, and we will provide basic care and basic diagnostics, physical exam, treat fractures, get fractures started so that they're not unrestrained. And then the duration of quarantine that's been very doable at each wildlife center has been quite variable. Mostly at our centers, we've really only been able to deal with a 24-hour quarantine. And then once the animal is warm and well hydrated, it gets screened again. Has it manifested neurologic signs? If so, maybe you'll get euthanized. And otherwise, it's going into the regular clinic care where it can get exams, x-rays, surgeries, whatever it needs. So it's kind of a different way for us to process animals. So here's our lovely PPE that we wear for new animal exams. That's our staff from our Los Angeles center examining a new pelican. We've been training our new protocols to volunteers. We did go a few months, thankfully, during the summer where there weren't really any cases in California. So we did loosen things up a bit, but in large part over the winter when our migratory birds arrive again, we're tightening things up again. And we have been imperfectly carving out quarantine spaces in buildings that were not designed for this. And every wildlife center has similar challenges. So we ended up with this is normally what we call a pelican box. It's a pelly box. So we turned a pelly box into an area where we could don and doff personal protective equipment. And then inside the building, we carved out this quarantine area. So there's yet another small box inside where people can go and watch an animal and try to decide whether or not it's got symptoms of this disease. And if not, then it goes into the quarantine area to get warmed and rehydrated and started getting treated. This is the outside of that in our general care area. So our facility definitely has lots of large spaces in it. It's designed for oil spills. It is not designed to have giant quarantine spaces. So we created some extra rooms, and that is certainly not perfect, but it's what we have. So
Becky, really quick.
Yep.
A question I think you would be better placed to ask. Jonathan Moore asked, how readily can symptoms of domoic acid poisoning be identified versus HPAI?
Well, the thing about domoic acid is that it's going to affect a large number of animals at a time. Like the last time we had a really big outbreak was in 2017 when we had loons. We had like 120 loons come in. So there were several hundred dead loons on the beach, but a whole bunch of them were alive. They were it. That would be a circumstance where we would immediately be testing them for domoic acid, and also we would have oceanic data available. So it's something where if we had a whole bunch of animals, we would be thinking, okay, which is it? We need to differentiate. It's just the same right now when we're dealing with a botulism outbreak in not only asymptomatic carrier species, but also a bunch of shorebirds and pelicans. So it's complicated and it requires doing some testing. If we're not sure, we can always spend the money to do some testing for avian influenza to try to rule it out. And we can also test for domoic acid, but it's a little more complicated than the avian influenza test. So anyway, we do the best we can to figure it out. Okay, so this is the classic clinical signs of transmission of highly pathogenic avian influenza viruses in poultry. So the incubation period is thought to be 2 to 21 days and can manifest all of these different clinical signs. So it tends to be respiratory or neurologic. They have some gastrointestinal signs like diarrhea, inappetence, weight loss, weakness, lethargy, etc. And then virus is shed in all of their secretions, poop, and all that. And then the infection can spread pretty quickly, not only from the infected bird, but also from any surfaces that were contaminated or their water source. So sounds pretty scary, right? And many of these clinical signs are pretty much the definition of what we get every day in wildlife prior to this disease. Like they're emaciated, they lost some weight, they're weak, they're lethargic, they have diarrhea. You know, that's like normal intakes for wildlife rehabilitation. So we had to be able to needed to be able to differentiate these. So this is what we've been seeing in wild birds. It's been largely neurologic, and there have been only a very small number of species, namely the corvids, crows and ravens, that have had a small amount of ocular and nasal discharge. One of the other clinical signs that's come up with these guys that was not mentioned in chickens is corneal edema. And I'll show you that in a video in a minute. The other odd thing, I might need to mute myself while I cough for a second. Sorry, talking too much. Most of the animals have been in good flesh. They are not skinny animals, which tells me that they're going down with the disease pretty quickly. They're not having time to become skinny. And let's see. So we don't actually know what the incubation time is in any particular species. Hopefully more on that will come out as time passes. So those have been the big, man, my voice is going suddenly. Hang on a second.
I'll jump in for a second while Becky maybe gets some water. Becky, you need to get a glass of water? Oh, there you go. Great. I just wanted to warn folks, if you are someone who is a little squeamish, there won't be any guts and gore, but the photos of the birds who are affected by avian influenza can be a little challenging if you are sensitive to that sort of thing. So I think it's important to see for those of you who want to. Becky's included quite a number of those. So we're going to go into those very soon.
Okay, let me try this. All right. So here we go, some videos. This one's courtesy of Patrick Hogan, who I don't think is on the video right now. Let's see. Oops. What do we do? Turn it on. There we go. So this is a gosling. Pointless motions. It's just kind of randomly kicking. Head doesn't seem to know what's going on. Here's another one. This is an adult. That's that corneal edema I was talking about. So their cornea looks bluish. The bird's just generally lethargic. Can't really stand. It's not very aware. Here's some more Canada geese. It's like nobody home, really. This bird seemed to have a head tilt. super weak, not very responsive. Is a different bird. These are all different birds. This one has some more of that corneal edema. It can be in one eye or both eyes. And then this guy's standing, but kind of acting odd. And once you look at him really close up, he's got little head twitches and stuff. Yep, so he's moving his head kind of rhythmically back and forth. Stafford. I don't know if you can hear Katie talking. So you can see those rhythmic eye movements. That's pretty typical. And then we'll get into some raptors. These videos are all courtesy of some other wildlife centers. So this is a peregrine falcon. This bird kind of looks weirder the longer you watch him. He starts to look like he's in stop motion. So he's doing some weird stuff with his eyes, got some rhythmic eye stuff going on, and he starts to like almost like a chattering motion of his head. So he's aware that he's being held, but he's definitely not behaving normally at all. He's doing that weird little head movement. Like, what's going on? Okay. This great horned owl is standing and fairly coordinated, but he's really out of it. Normally, you would not be able to grab a great horned owl's head. So standing, but unaware of what's going on. It's almost a little bobblehead going on there. And this is a turkey vulture. This bird looks weirder the longer you look at him. So at first he looks almost like a dead vulture, and then like, oh, with pestering, you kind of get him to respond a little bit. And then a little bit later, he starts to be weird. Okay, major head tilt. Very abnormal looking bird.
Becky, another good diagnostic question. This one's from Jill Brennan. How can you differentiate the neck weakness from botulism versus avian flu?
So one of the serious differences between the two is a lot of times botulism birds don't look like they have a problem in their brain. You know, it's kind of like front end, back end problem. Botulism tends to be like overall weakness, and but they don't look like they have encephalitis. Like they don't look like they're not in their right mind. They just look like they're super weak and can't stand. So you might notice that some of these birds can stand fine. They just don't know what's going on because their head is having a problem. So I'm kind of differentiating it that way. Let's look at some more of these. Here's a Western gull. This is a bird that was found floating in Pillar Point Harbor on San Francisco Peninsula while there were thousands of brown pelicans perched on the rocks. And this bird was captured by a friend of ours who spots our blue-banded pelicans all the time. So he caught that bird, fished that bird out of the water, carried him in his kayak for quite a while, and then disinfected his kayak. After I warned him that I think the bird had avian influenza. So here's a brown pelican. This is the only brown pelican case that we have had come into our clinics. There have been a couple other cases in California. But this bird is just, this is the same bird in both videos. Let me play them both. So we turned him around. So he's also able to stand when sufficiently pestered, but he's holding his head really weird. You can't really tell what's going on. He's doing some weird rhythmic eye movements. He's also sort of awake, but like, oh, doesn't he? He looks to me like he has a really bad headache. Like, oh God, I'm gonna go to sleep now. Even though he's standing up. So they're definitely acting like their heads are a problem. Okay, sandhill crane. This guy is a little more jittery, unable to stand. He's got a little head tilt, and he's doing a lot of jittery stuff. Snow geese, we got a ton of snow geese. We don't normally get snow geese very much, and we had like, I don't know, 40 of them or something. Not all of them got tested, just a couple, because we certainly can't afford the testing either. This bird could not stand at all, even though we pestered him quite a bit. And he could stand for just a second, and then he's like, oh, I'm going to go press my head against the wall again. Head pressing is kind of a sign of encephalitis in a lot of species. And there he goes. He's just going to like prop himself up against his head. But, you know, think of it kind of like yourself when you have a really horrible headache, like pressing against the wall might feel good. It's black-crowned night heron. This was at our Los Angeles center. You know, this bird might be car hit, might have 3 fractures, you know, who knows? So we are physically examining all these animals and trying to figure out, do you have a musculoskeletal reason to be like this, or are you otherwise uninjured? So anyway, what we've seen largely in all the centers I've talked to and in our own hands is that the animals largely act like they have encephalitis. They have a brain inflammation, they've got tremors, they can't hold their head normally, they're uncoordinated, they might be circling or head pressing or having a seizure with lots of tics and twitches and subtle tremors. They might look bubble-headed, like they're just like standing there moving their head weird. They might seem like they are have a reduced reaction to stimuli, or they might be even barely conscious, no matter how much you bug them. Some of them may be standing but like nobody home. Corvids are kind of like that with West Nile as well sometimes. So it would be difficult to differentiate a crow with West Nile versus avian influenza, but neither of them have a good prognosis. They might be unable to stand up or even right themselves. These paddling, pointless motions, weird eye stuff going on, rhythmic eye movements in any direction with that corneal edema. And then there have been a few cases where people have sent us videos of Canada geese walking, like even walking with their eyes closed, like just walking around blind, blumbling into stuff. With the corneal edema, it seems like it's one of those clinical signs that's fairly diagnostic, and kind of like the loss of taste and smell, I suppose, with COVID. So similar kind of a thing that came out of this. But it's not a generalized weakness. It's not hind limb paralysis, paresis like botulism. Okay. So lots of challenges to dealing with this. It is highly lethal to a lot of species. We don't really know how contagious it is. All we have to go on is poultry, really. But hands-on experience is kind of telling us that it doesn't seem to be as contagious as we fear within wildlife centers. Talk about that more in just a moment. We have no idea whether there's effective treatments. So far, there have been some positive California condors that were treated and released. At least 2 of them have been released. They were treated with Tamiflu, which is normally an anti-influenza drug reserved for humans. You know, there's always a fear if you use a drug in animals that it might not be effective in people anymore. So it's like, we use this in animals. And Minnesota Raptor Center did release 1 great horned owl that had a mild case and appeared to recover and tested negative. So there is definitely some zoonotic risks with this disease. So dealing with it in our facilities where we have inadequate quarantine spaces, and then you need redundant staff, it's making treatment attempts would be very difficult, which is not to say that we would not do it in the future, but we certainly might. And then we have to deal with those asymptomatic carriers having risks to other patients. It's definitely difficult to differentiate from some of these treatable neurologic conditions that we talked about. There's no point of care tests. So remember COVID, before we had the at-home tests, the poultry rapid tests, unfortunately, mostly give unreliable results for wild species, and they're not currently recommended. And then the send-out tests to our state lab in California are pretty expensive at $75 each. There also is a pretty long time lag for definitive results. We're lucky in California that our state lab, despite the expense, they give us really awesome results really quickly. But then final results have to go through USDA's lab, which also has had some odd things where all of a sudden USDA and figures you had a positive bird, and then 8 weeks later they call you and go, how are you feeling? Who handled that animal? You remember that bird 2 months ago? I've only had one of those calls, but I know our staff has had a few calls. And then each county, even within California, has been acting very variable about this. Los Angeles County is right on it. All the other counties we've been dealing with, I've heard San Diego County is right on it as well, where they're actually really trying to help. But some trying to do human contact tracing for every positive bird. But a lot of the health departments are like, what? Very disinterested and clueless. So we have some overarching concerns about this disease in wild birds. It is yet another hit to wild bird population health among all the droughts and fires and habitat loss and reduced food that they have available. And you know, as animals get crowded more and more into reduced habitat, that increases the spread of disease, right? Because they're in close proximity to each other. So there are a whole lot of issues that animals have to deal with. And this disease may push some vulnerable species that are already in trouble, threatened and endangered species, over the edge to extinction. Totally might happen. And it also might drive some species that are currently in fine shape into threatened and endangered status. And unfortunately, the disease is probably not going away. This particular strain has been circulating quite a while now in Africa and Europe, and it's probably not going to just burn itself out and go away. Like it's still in its, I don't know, it's still wreaking havoc all over the world, except in Australia and Antarctica. So our lessons learned so far, wildlife centers in the US, we have not really had a problem with in-house spread. And I was part of a symposium talking in New Zealand recently about this disease, and a researcher from the agencies that deal with poultry in Great Britain spoke, and they have done some studies over there with this disease, and they were finding that it was not especially airborne. It was largely coming into chicken houses on water or roof leaks, those sorts of things. So it does appear to be that it's more direct contact with feces. And within wildlife centers, we typically don't have animals in contact with each other's feces until they're in an outdoor aviary well along the way to being released. So it might be that our ordinary practices are helping prevent some of the spread. But our screening and quarantine protocols, even though they've been pretty good, we're also probably euthanizing some treatable animals, which we would like to stop having to do. We've definitely learned, and some states have not learned this lesson. So there are some states that are forbidding people from taking ducklings, et cetera, not allowing the rehabilitation of some of these animals. And unfortunately, that doesn't mean the public is going to have those animals euthanized. Like mostly people will find a sick animal and they want to help it. So people in general are going to keep those animals if there's nowhere to take them. And they will put them in the backyard with their chickens, so it doesn't really help anything to prevent people from caring for these animals. We certainly know that that's the case with oiled animals, where this dude was from an oil spill in 2015 in Santa Barbara. He didn't think the state was acting fast enough, so he had to take that pelican off the beach. We desperately need reliable point of care diagnostics for wild species, and a lot of research is needed for that. So what can we do about this horrible, horrible disease? One thing we can really do is reduce the other threats that all of our wild bird species have to deal with, like increase their access to decent habitat. So cut down on habitat degradation and loss. Continue getting rid of fishing gear interaction problems. That's a big killer of wild birds, especially the water birds we deal with. Introduce predator reduction and elimination is always a great idea to help animals out that are not supposed to be dealing with that predator. Looking at you, kitties. Overfishing is a big problem, so that animals, especially the seabirds that are so at risk from this disease, have enough to eat. And if, probably the best case scenario is if those wild seabird colonies that are being really horribly impacted by this, maybe they'll have a couple generations where their chicks have increased survival because there's enough food to go around, because there aren't very many of the birds. So hopefully there will be excellent juvenile survival to try to replace some of those animals. But unfortunately, seabirds tend to have a really slow reproductive rate, like only 1 or 2 chicks a year. So even the survivors may take a long time to replace their missing conspecifics. So we can reduce toxic spills and oil and all that sort of stuff that really causes birds to die. And even some of the things like, windmills and window designs can really help with some of those raptor deaths, kind of offset. So the more people can do to reduce these unfortunate human-caused deaths for other reasons would really help a lot of these species. Our threatened and endangered species may be able to be vaccinated. Obviously an animal has to be handled, and what's the duration of the vaccine's action. So with some species like California condors, maybe that's possible. Obviously that wouldn't be possible with hundreds of thousands of animals. So it's a limited application there, but nonetheless to be considered. And then some of these species of animals have biologists that professionally monitor their populations, but there are a lot of organisms in the world where nobody's paying attention, or they're not paying attention very often or very much. And if animals die in droves in the wild and no one notices, there's nothing we can do about it. So supporting biological monitoring is really important, making sure that gets to be a budget line item in some governmental projects and all that sort of stuff. And then from the public point of view, if you see wild animals in distress, do something. Either reporting dead birds to your state and local authorities, or calling your local wildlife rehabilitation center for live animals, and then rescue them if you were advised to do so, depending on the circumstances. And then supporting your local wildlife rehabilitation center can be really helpful. And it's also, it helps save animals one at a time. We also, of course, want to make the animals that we release go out there and be successful animals that have lots of babies and help replace any animals that have been killed off by illness or habitat problems, et cetera. And local wildlife centers certainly need funding wherever you live to support all these actions to support these species. So what are we doing at Bird Rescue? We're just about to start a research project with UC Davis evaluating the utility of certain point of care tests to try to figure out whether we can have a point of care test that's useful and effective in the different species that we treat. We're also continuing our investigations and work on other health issues that our animals have. Like we're involved in a big DDT legacy contamination project off our Southern California coast. And we, of course, our standard animals that need to come into care have a lot of fishing gear injuries, or they've been hit by a car, that sort of stuff. And of course, oiled birds is something we treat routinely. And then in the future, we hope to be able to assess potential treatment options and feasibility for some of these species. And then we, of course, need funding to support all of this. So I've got a whole list of some of the really interesting papers on what's gone on in the last year or so. For example, in Peru, that second one there, the first one's talking about avian influenza in the northern gannet population of the North Atlantic, talking about all the breeding colonies all around the Atlantic, which is a great big picture paper. That also includes some of those pictures you may have seen on the news of the different iris color in some of the animals that seem to have recovered from the disease. So these, all of these websites, we can probably send these out as a PDF or something if anybody wants to dig deeper into any of this. It's all really interesting stuff. Some of these papers might be a little heavy on the science, but there's certainly a lot that you can get out of it anyway, if you're not a science-minded person. And that's about all that I have for you. So thank you very much for your attention. Sorry, my voice is a little weird tonight. And I'd be happy to
Hey, Becky,
answer
another
any
question. Yeah, another question. And I would love to invite folks to raise their hand if you don't want to type them in. But this is another one from Jonathan Moore. Is the point of care test research focused on immunoassay, like lateral flow ELISA? Those are all words I don't know.
Actually, we're working with a PhD student from Davis, and it's an in-house PCR. So that's going to be cool if it works. It's called from an company called Biosys, if you want to look them up. Anybody else?
Request a yes on the request or the offer of a PDF of the research. So we'll make sure that we put that where folks can get at it. Thank you for all your work caring for the birds. I am just looking to see, I don't see any hands going up right now, but the Q&A portion is going well. I've been answering a lot of the questions quietly in the background.
Well, looks like David's not here. I was going to ask him to give us an update on what's going on in South Africa, but I don't see him. He's on the original list. But,
Time zones.
I know.
I think a lot of people signed up because we are recording, and we will send out the link to the recording later on.
Questions? I know. I like somebody's Susan Kirk's advocating for discontinuing large poultry operations. Transitioning to plant-based nutrition, advocating discontinuing wet markets. You know, ultimately, some of those issues, I should look at your answer. Yes. I was just reading a science fiction book where one of the premises was a horrible disease that made it so that the poultry industry, like we couldn't eat chickens anymore. Which was kind of an interesting take on a book. It's like, did you know we were going to have an avian influenza problem? Author who wrote this 5 years ago. But yeah, large poultry operations are a huge problem. They really are, because they're like disease factories, frankly. And I love eggs, I really do. But I would gladly trade ever eating them again for not having this problem. So it you know, yeah. There's also too many humans on the planet, so there's that issue too. But nobody really talks about that one.
Saw a hand for a second. Susan McCarthy, I'm not sure if you were trying to or if that was an error. There is a new question. Becky, I think you can see them yourself, yes?
Yes. Do you expect
Great, if you don't mind.
governmental funding for the wildlife rehab centers will increase and start soon? What is needed to accomplish this, in your opinion? Thank you for sharing this ingredient from the Netherlands. Oh, hello. We have a tiny bit of governmental support in California, but only a little tiny bit. And I don't think anybody's going to be giving us any money anytime soon. Do they give you money in the Netherlands?
Yeah, I started off the talk with pointing out that you know, when it comes to animal issues in the States, you know, animals we eat, animals we hunt, animals we harvest, those are the priority. In fact, sometimes those are the only animals managed. And actually, to Becky's point earlier, when you see a sick animal, it shouldn't be assumed, depending on where you live, that the animal control or the public agency will actually be able or willing to help out. So some of even the surrounding counties here in the San Francisco Bay Area where I live, quite surprisingly, will not respond to anything but domestic animals. So it is definitely an ongoing challenge, and something sadly we are in the minority who love and try to take care of wildlife.
Yeah, I mean, California has a checkoff thingy on your taxes where you can donate to a fund that gets distributed to the wildlife rehabilitators based on how many animals a year you deal with. So that is a tiny little fraction of a thing that that provides a little bit of support, which is great. Better than zero, for sure.
And I will say that you know, Bird Rescue is very fortunate to have a strong supporter base, which allows us. I think many rehab organizations would die to have the capacity of someone like Becky on their staff. So we're really grateful that supporters allow us to fill in that gap. Linda Morgan, you are raising your hand, and I believe I click a button to allow you to talk. Go for it. It looks like you are muted, so I'll ask you to unmute. There you go.
I'm looking at the picture here at the end and wondering what was the occasion for all those pelicans down at the Southern California facility.
I love that picture. Kylie took that. Kylie Clatterbuck, our center manager down in LA. That was during the brown pelican mass training event we had in May, June 2022, when we had way too many pelicans in the aviary, but thankfully they're sociable birds and get along.
And just one white pelican amongst
and one
a
white,
lot
yeah.
of brown ones.
Noticed that.
Looking at his leader. Yes, love that photograph.
Another question coming through the Q&A, and thank you, Linda. Really nice to hear your voice. It's been a while.
Yes, hi, Linda. Strange that you didn't see very many brown pelicans in California, as we saw a bunch down here in South Florida. Yeah, we didn't. And it seems like our breeding season of our California brown pelicans on the Channel Islands went off without a hitch, I think. At least we're in touch with most of the biologists that monitor out there, and they did not report any nasty, scary things happening. So they may have escaped it. Now, thankfully, our California brown pelicans do breed in a more deserty kind of location, and that could be part of their protection, hopefully. Since it's a little drier down here. Do you work with
Becky?
San Diego Zoo Wildlife Alliance for testing research, for instance? No, we have not. Do you have a contact? Do they want to run tests for us? That would be lovely.
Jonathan Moore would like to speak, so I'm going to allow to talk and ask you to unmute, Jonathan. Jonathan, go ahead when you're ready. Oh, I'm not sure if your sound is working. We can't hear you. It looks like you are unmuted and you are allowed to talk. Zoom says. Might have to go back to typing the answer.
Yeah.
Sorry, Jonathan, we can't hear you for some reason. Nope. Maybe go ahead and type your question, Jonathan, and we're happy to cover it. We've got about another 10 minutes. We don't have to go the entire time, but I want to respect the fact that poor Becky is losing her voice.
Leslie Straub asked, there was no funding available for testing to help with monitoring. So the California Department of Fish and Wildlife has a certain amount of budget that they were able to devote to this, but it was really within their ordinary budget for all wildlife diseases or all avian diseases. And they primarily only have one staff person who really deals with that. So it, there was some testing with that, but if you have a live animal, you don't know how long to quarantine, having an immediate test is really important. You can't wait forever for test results to come back, and keep every animal separate from every other animal. So yes, a little bit. We have been somewhat fortunate in the LA County. The county public health decided to up their own testing thing, and they will allow us up to 5 freebies a week. But again, it takes about a week to get the results back. And it's certainly not an immediate good answer like the state lab that costs so much more.
Great. I think Jonathan was just trying to tell us that he doesn't have a specific contact for HPI testing at the Wildlife Alliance.
Mm-hmm. Let's see. Vaccinations being trialed with condors, something trialed in your birds. So in the United States, I don't know where you're from, but all vaccinations for wild animals have to go through USDA and get approved to be used. And it's something that generally will only get approved if the animal is essentially endangered. So it's possible something like that could happen for the western snowy plovers that we deal with if they started having a big bad problem with it. But most of the species we deal with are not as endangered as all the condors.
Great. Well, while folks think of maybe their final questions, I really want to say thank you so much for joining us. Again, the goal was to give you a high-level view on bird flu. We really do want to help lift the voice, and please do share this information with friends, especially folks who have backyard chickens or ducks. That is a really big concern, of course, crossover. And Becky, thank you very much for making the time and for taking my push to simplify as much as possible what is not at all a simple topic.
It's really hard to simplify stuff like this, unfortunately. If anything, that was simplified. That was the simplified version.
We're going to do a test now. Which one was shaped like a bullet?
Yeah, right.
All right. Thanks, everybody. Have a great night.
Thank you.